organizations, I'd like to welcome you this evening and thank you for coming. This is the second in a 5-part national series on trust in the new sciences, Remaking the Human, which is organized jointly by CCEPA and the SURE-funded National Cluster for the Humanities and Social Studies of Science, or Situating Science, the Knowledge Cluster. CCEPA is a joint venture of the Atlantic School of Theology and St. Mary's University in Halifax, and works to provide an arena for critical thinking on issues where ethics intersect. This national lecture series is also greatly supported by the local node of the Situating Science Knowledge Cluster here at UBC, headed up by Dr. Alan Richardson, who has been hugely instrumental in hosting tonight's event. I'd also like to acknowledge the support of the Science and Technology program here at UBC, as well as all the on-site assistance we've been having in university, and also a partner in the Situating Science Knowledge Cluster. I'd like to add just 3 small things. Firstly, I'll call your attention to the evaluation forms that you see in front of you. Your feedback is really important to CCEPA because it helps us in the development of future programming. So if you could take a few minutes just to fill that out, we'd greatly appreciate it. We'd especially like to know how you came about to hearing about this event and whether you come from an academic or a public sector. Secondly, tonight's event is being videotaped for DVD production. So I'll ask those of you that participate in the Q&A period to follow, which will follow the formal part of this presentation, to use one of the aisle mics, which we have set up here at the front. I'll also let you know that tonight's event is part 2 of 5. And that doesn't mean you missed anything thematically if you didn't see part 1 or don't see 3, 4, or 5. But it's to tell you that these productions can be viewed and downloaded both on the CCEPA and the Situating Science websites. Part 3 of this series goes forward in Toronto in early March, in Montreal in later March, and in Edmonton in April. Parts 4 and 5 will also be reprised in Halifax in March as well. All these productions can be viewed on those websites that I mentioned. And finally, I hope you'll join us after this presentation for a reception that we'll host in the lobby, and we can continue this conversation. So now, please help me in a warm welcome for Dr. Leslie Norman.
Thank you very much, Chris. On behalf of the Situating Science Cluster, I'm pleased to welcome you to tonight's lecture. As you can see here, Science Friction, Personalized Genomics and the Future of Medicine. What I plan to do right now is that I will be giving a short introduction of both of our speakers and then a full introduction of the first one. And we will have the longer talk by Michael Hayden first, followed by the response from Anita. And then I hope that it'll be very controversial and they will want to debate one another. And especially that all of you will want to leap in there. So I will be moderating to make sure that as many people as possible can have their chance to talk before we've completely exhausted ourselves and we feel that we've earned our reception at the end of this. So let me tell you that our tonight's speakers will be, first of all, Michael Hayden, medical geneticist, followed by Anita Ho, an applied ethicist. Michael Hayden is a full professor of medical genetics at the University of British Columbia and the director for the Centre of Molecular Medicine and Therapeutics in Vancouver here. Dr. Hayden has served as the Chief Scientific Officer for Genome Pharmaceuticals Incorporated since March 1999 and has been a member of the board since March 1996. He's the author of over 400 peer-reviewed publications and 150 invited submissions. Dr. Hayden focuses his research primarily on genetic diseases. Dr. Hayden's work focuses on understanding the genetic roots of illness and understanding and using that understanding to develop better approaches to treatment for patients. He researches diabetes, coronary artery disease, and is part of a large collaboration to determine the genetic basis for adverse drug reactions in children. Much of his career has also been dedicated to understanding the development of Huntington disease and finding a way to cure it. Dr. Hayden is currently conducting research into the genetic factors that determine differences in drug reactions between individual patients. A topic of his discussion here tonight will illuminate this. Dr. Hayden is the recipient of numerous prestigious honors and awards. He was elected to the American Society of Clinical Investigation in 1992, the board of the American Society of Human Genetics in 94, and the Royal Society of Canada in 95. In 2007, he was awarded the Prix Galien, one of the highest honors in the field of biopharmaceuticals in Canada. He has also received the 1998 Distinguished Scientist Award of the Canadian Society of Clinical Investigation and the 2000 BC Biotechnology Alliance Award for Vision and Leadership. More recently, he was named Health Researcher of the Year in 2008 by the Canadian Institutes of Health Research, and in 2009 he was named to the Order of British Columbia. I was going to say the British Empire. That's the next one, obviously. But you're not here to hear all about this, which probably makes us all tired just hearing you give this introduction, but rather to hear Michael Hayden speak. So I will turn the floor over to Michael Hayden. We'll be talking about what are we waiting for, personalized medicine and trust in healthcare.
Thank you, Leslie. Thank you, Chris. It's really wonderful to be part of this national series spanning the country. And I think the topic that we're going to address tonight is one that affects all of us. And I just want to also start off by giving a very special thanks to Mike for helping me and aiding me as an emergency to provide me with this wonderful pointer. So Mike, thank you very much. These are the students supporting the professors. I hope we can reciprocate. So when we think about genetics, genetics is everywhere, and actually Vancouver's had a special role in the Human Genome Project. In fact, it was the year 2000 where the first draft of the Human Genome was announced here in Vancouver as part of the World Human Genetic Conference in the year 2000. Shortly thereafter, this was announced by Francis Collins, now the leader of all research at the NIH, and also in the presence of Bill Clinton and also Tony Blair. And at the time, we were all filled with incredible hope, optimism about what this could bring. This was, in a sense, a miracle. What was initially predicted to, in 1990, to cost $3 billion in about 15 years, was finished a little ahead of time, but did cost close to the $3 billion. And as you can see, this was a wonderful map. And the hope was that this map was going to lead to new approaches to treatment, new ways to diagnose, predict, and prevent illness. And we were all filled with much optimism. In fact, Prime Minister Tony Blair at that particular time said that cancer will soon be a thing of the past, and the gene code was going to beat all disease. So this was a time of hyperbole. The scientists joined with the politicians in a chorus of hurrah. And in a sense, some of us, whilst having some skepticism, were still very hopeful about what could be achieved in a very short time. After all, at this moment, we had the DNA sequence that defined who we were. After 2 billion years of evolution, we were able to define all the letters of the alphabet that made each one of us. And with that alphabet, we learned many things about ourselves. We learned about a relationship to every living species on Earth. We recognized how related we were to a grain of rice, to a caterpillar, to a worm, to the monkey, and importantly, to every other human being on Earth. We were, in essence, brothers and sisters, linked by our genetic code. And at the same time, we also learned about how incredibly different we were. We recognized that there was nobody like us on Earth. In fact, for all of you here tonight, and for all of us on Earth at this very moment and forever, there's never been anybody like you, and there never will be anybody like you again. Your DNA is unique to you. So at this point, this was really the culmination of much hope, much hype, and I'm going to share with you some perspectives about what's happened since that particular time. Since 2000, or over these 10 years, where are we? And at the time, there were many who predicted that this was going to change the way we viewed the world. More important than putting a man on the moon, medical nirvana is what Francis called this, and Francis, a colleague in this field, and now in a position of great influence in the United States in terms of influencing health policy and also health funding. And in fact, this is what Francis said. And this is relevant because, of course, we've arrived. So we can say, okay, by the year 2010, we're there now. Where are we? So this is what Francis predicted. There would be predictive genetic tests for many common conditions, and primary care providers will be practicing genetic medicine on a daily basis. Well, what a pity that almost none of what was predicted has really come to fruition. And the question is, why not? And why were we so wrong in those particular predictions? Now, of course, Francis made these predictions, and we all joined him, by the way. It's not just Francis. He was a lead of the Human Genome Project. Many of us joined that chorus, some with some greater enthusiasm than others. And in fact, the press became an important partner. And so when you think about how science reporting works, generally this is how it goes. So if we tell a reporter, and in fact I'm part of a press release that's going to be released at 9 o'clock tomorrow morning, and again, talking to the press is something we have to be very careful on. So here we say we destroyed 10% of cancer cells in a lab rat's tail, and the headline might be cancer cured. Or if we didn't say we didn't cure cancer, but we're just moving faster towards a future treatment, time travel discovered. So the press is really also a major partner to this, and this is really the science news cycle as reported in the Scientist. So if we look at, and much of our work is correlation, I say this is associated with this, and this can be used as a proxy for detecting this particular disease. So I might say that there's some association between A and B. Now the problem is university PR officers, their role is to publicize this research, is to really publicize the university, raise the image of this university in the public. So the immediate release would be scientists reveal potential link between A and B. The newspaper reporters also at this point have really got it, and the science reporters, and there are, by the way, fewer and fewer science reporters there. If you look at what's being cut right now in terms of the news organizations, science reporting is often the first to go, which is a real problem. But they would say A causes B. And so it goes on, and then A causes B all the time. The local news, what you didn't know about cancer can kill you. And finally, you can see, you go to see your grandma, and she says, I'm wearing this to ward off A. So the cycle is one, even if the scientist talks about a little linkage. And by the way, if you look here, A is correlated with B. You look at a p-value, which is a statistical value, that means it's absolutely not significant. It happens to be associated, but there's absolutely no significance. And so this translation of the science news cycle continues. It starts with the scientists, is fed into by the universities, the press continues this, and then when we come to the interviews, we actually also participate in raising this particular specter. And so, of course, with all of this, the view has been that everything is caused by genetics. In fact, everything is genetic. And everything has to do with your genome. And when you look around, you look at really your family, you believe that everything is genetic. And so genetic fundamentalism has played a role today in the view that discovering our DNA means that everything is caused by our DNA. In fact, even in Canada, hockey, how you do in hockey is in the DNA. And if you go on further and you look even about our religious beliefs, there's now a God gene. And if you go on further and you look in New York right now, there are these parties, where in fact parties where people get together and they want to decide, really learn more about whether their children should be involved in certain kinds of sports. And this is actually happening. It was reported in the New York Times. It has to be true. And so if you want your DNA mapped, you can actually spit this out and you can have it mapped, and you can then know whether your children, and then also get children mapped, and you can determine whether your children have a certain allele, a certain type of DNA that predicts whether they're going to be fast in certain sports. When you look at the association for this, as has been reported, the association is a bit like I told you before, marginally significant, not all that predictive, and yet this is what's happening. And this is being exploited, again, by commercial companies in an effort to really offer this to susceptible and vulnerable and also embracing parents in an effort to give their children an edge in terms of that sport. And so this is not what I would call genomics and genetics that's relevant for health. This is very much recreational genomics, and people should be free to be able to follow whatever they want in that regard. But this really is not what I'm talking about when I talk about the impact on health and disease. What we are interested in doing is defining whether these genetic variants can do anything to help us predict whether we are going to get some serious illnesses, and then if we can change our lifestyle or change something in an effort to moderate that risk. That's really from prediction to prevention. And again, there's been a lot of hope about this. Diabetes, are you at risk? Well, in fact, we know that a large proportion of the population, 1 in 5 people by the age of 60, is likely to develop diabetes. And I show you this only to show you that this is published in very, very serious journals. And here's the odds ratio. An odds ratio means what's the likelihood that you will get it if you had that particular variant in your genetic makeup. And you can see when you start off with 1, so this is a 14% increase, this is a 22% increase, and that sounds quite high, until in fact you start recognizing that if your risk starting out is around 4%, and you have an odds ratio of 1.18, your risk ratio has now gone up to 4.7. So there's no, even though it's close to a 20% increase, it's really not a very dramatic increase. And clearly, this really is not predicting with any certainty whether you are going to or not going to be able to develop this disease. So many of the genetic markers that we have, whilst published in major journals, offer very little predictive value. Small increase in risk, small changes, very wide confidence limits, and actually very uncertain results. And here's another example. Height. We always say if you want to determine how tall you are, just look at your parents. My son, for example, looked at me and immediately wanted to disown me because he was interested in basketball. And the truth is, however, when you look at specific genes, you realize these genes, and this is a major study, 16,000 people, and people always believe that if you do a study, you have a small odds ratio, small increase in risk will increase the number of people you study. And here are people studying 12 genes in 16,000 people, and you realize this is just accounting for 2% of the variation in height. And if one person had all the genetic variations for height, they'd be barely an inch taller. So I think what we're saying is that it's much more complex. Yes, we can define certain genes that contribute a small amount to this, but the impact on this is rather small. And if you look at all the studies, and these are hundreds of millions of dollars now gone into these particular studies, interesting from a scientific point of view, but truly from a predictive point of view, what you see is that most of the studies have had risk ratios in this particular range, less than 2. In other words, there's no doubling of risk. If you start off with a risk of 1%, your risk is now 2%. Well, it is an increase, but still there's a 98% chance that, in fact, you won't get this. And how do you live your life if you have a 98 versus a 99% chance? Very hard to moderate your lifestyle based on a small difference in risk. What we're interested in from a clinical point of view is clinically relevant risks, risks that have significant impact, risks that are going to dramatically impact on your risk. And these are risk ratios that are right there. But you can see of all these studies, large numbers of studies, only a small number are in the high risk range. So when we say your life is in your hands, yes, for a small number of genetic risk factors, for most of them, in fact, the instructions for the personal genome age are quite confused, quite difficult to interpret in terms of your own life. Very interesting from a scientific point of view, because they give you insight into pathways, they give you insight into new thinking about mechanisms, but they're not ready for prime time today. And I just want to give you one more example that this is a recent one. This is October, just last year, where there was this new variant associated with Alzheimer's disease. And this was, and you looked at this risk ratio, the risk ratio was in the ones again. And yet when you saw this, let's look at how this was reported again just very recently in this particular issue. So here's a genome-wide, it's a study of variants, and what did Professor Julie Williams said? She said the discovery of these 2 new genes would provide valuable new leads in the race to build treatments and cures for the devastating condition. And this was an author that wrote this manuscript from Wales. First author was from Wales, 25 authors was everywhere else in the world. But this prompted the First Minister of Wales to say a major breakthrough in the battle to understand, develop treatments, good news for 7,000 people in Wales, etc., etc. So the hope, the scientists led this was grabbed on by the politician, and the hype continued. And so when you look at this, you wonder, what was it? What was it when you look at and you predict, what was it that was really precipitating in that particular instance, these people participating, and again, in obvious hype? Well, of course, there are many pressures. The line of funding in terms of research is going down. People need to differentiate themselves as scientists from the other people seeking funding. Politics, the line of politics also goes down. And if you look at various public approval and the line of career advancement, these are all kinds of machinations that are influencing these particular processes. And so the overpromising is driven also by the media that are asking the scientists, when they come to me for a message, they say to me, don't make it too complicated, make it very simple, and you only have 8 words to give the message of the last 10 years of your life in terms of your work. So make it definitive, and if you can, make it sensational. And of course, and then often from a scientist's point of view, if you're talking, for example, about the stem cell debate, in the DNA world, there was, again, many debates. You need to, basically, you're trying to overcome public opposition to it. There are sometimes commercial associations, and then funding issues plays a big role in terms of why we're doing this. And if you just look at this, the Danish Prime Minister thinking about policy and speaking to the scientists such as myself, saying, don't provide us with too many moving targets. It's already very, very complicated. I need targets, certain figures, not fit too many considerations, uncertainty and risk and things like that. So the politician's saying, just make it simple. Just make it simple, because that way I can get the message across. So it's not just the scientists. It's the milieu in which we're working. It's the media. It's the politicians themselves that are looking for this. However, when you look further, this was what was also reported on that particular, this was the Sun in the UK, spoke about it, the biggest advance in Alzheimer's research for 15 years. What had been discovered was some association with a gene that really gave you some indication of a pathway, but had very little predictive value and was not a genetic cause for Alzheimer's. Then Dr. Roses, and now you see the competition. Dr. Roses was the guy who discovered one of the early genes for Alzheimer's. He said, well, these 3 genes, really minor effects, which they did, compared to the variant side near EPA-E4. Well, guess who discovered that one? Okay, Dr. Roses. And their biological role in the disease unclear, despite the statistical data. So, and this is really, so when we think about DNA, this is what this week's Time magazine cover looked like. And all of a sudden, why your DNA isn't your destiny. So there's a shifting mood. Your DNA is no longer seen to be totally predictive of your future. There are other factors involved. And now we realize that it's not just about the DNA that's important, but rather the environment can influence the way your DNA works. Because your DNA encodes in messenger RNA, which is a message that is translated into a protein. Essentially, everything we are is a reflection of our proteins and how they're expressed in different cell types. And we now know that, in fact, DNA, therefore, has to be transcribed into a message. But how that transcription occurs is actually influenced often by our environment. So, for example, if you put a person into a good environment, there may be changes in the expression of certain genes compared to a bad environment. So all of a sudden, the nature-nurture discussion that we've always held on to as if they were 2 worlds, separated, never to be linked, in fact, are linked through the world of epigenetics. And so we have the same genome our whole lives, in a sense, but as we get older, our epigenome changes. How genes are expressed changes, and that's very much influenced by our environment. So these are essentially changes that occur, but they do not involve changes in DNA sequence. It's no longer in the sequence, but the patterns of the genome that are influenced by how they are expressed. And certain things we know already influence the epigenome of a child now. For example, during pregnancy. As mother eats, and nutrition has big impact on the epigenome of a child, such that children born of mothers who've been very starving or in terrible conditions, such as during the Second World War, for example, concentration camps, and children were born during that period, these children might be more likely for various diseases in later life because of the changes not in the DNA is the same, but how certain genes are expressed from early life. And now we recognize that identical twins are no longer that identical. In fact, everybody who has twins and knows twins knows that they might look identical, but there's such differences in them that are recognizable by people close to them. And we realize now that these twins have similar DNA sequence, but they have tremendous variation in the way the genes are expressed, reflecting the change in the environment. So the identical twins have different changes. And most really wonderful, we've now learned that just rearing, maternal childbearing, and actually rearing of your children during the early years of life has big impact on their epigenome throughout their lives. And so it's not just that it feels good because it alters chemistry and actually alters the lifelong prognosis for offspring of children that are appropriately reared. And so the importance of dealing with poverty and dealing with some of the adverse environments isn't just social. It has major impact on the medical output and the medical outlook for these children for the long term. So childhood poverty is not just a social issue and not just an ethical issue, but in fact is a major issue on health, because the health of these children as they become adults will be altered by virtue of the environment they've had. So I think we're learning as we've gone to 2010 that it's not quite as simple. It's not just a difference of DNA sequence and they may be able to determine who we are, where we are. We're more complex, we're influenced by the environment, and even though we have this DNA sequence through life, we have multiple epigenomes. Some of them are inherited, but much of it can be influenced by the environment as we grow older. Now, there's some areas where, in fact, personalized genomics has dramatically revolutionized medicine. And in this area, all the hype is worthwhile and appropriate. In the area of cancer, we're now recognizing that many cancers are not just breast cancer, for example. It's not just one disease, maybe 40 or 50 diseases. And each of them is characterized by a new topography and a new anatomy and a new biology defined by patterns of gene expression in that cancer. And furthermore, if you define that, that'll lead to different approaches to therapy. So now we're recognizing early on if we can define the nature of the cancer by virtue of its genome, by virtue of defining the way genes are expressed, that will influence therapy. So we don't just treat breast cancer as breast cancer. We don't just treat a patient in that way, and particularly in cancer. So there's tremendous amount of information in terms of the multiple subtypes here of breast cancer. As you can see, HER2, for example, is a particular type of breast cancer that is particularly responsive to a drug termed Herceptin. And so you're able to classify. In the old days, you had a cancer and there was therapy. And today, we have a cancer, we classify patients, and we treat those with a particular disorder, and then we're also looking for stratification of patients to be treating the particular patient. This is the ultimate personal genomics. You have this disease, you're able to treat that particular patient, and as we define this, more and more diseases are being stratified, and so we're crossing boundaries, we're writing a new anatomy. It's the new functional anatomy of cancer, the new functional anatomy of different organs, and how they're perturbed by disease. And in this particular instance, this is often predicting how they might respond to therapy and in that way preventing this. So there's a new paradigm that genetic medicine is leading to. It's not quite there yet. Our current paradigm is still making a diagnosis and treating the patient. But we are looking to a new preventative paradigm where, in fact, you can take preventive action by looking at a risk profile. In this instance, you may be able to have an earlier diagnosis, better treatment. And for a few diseases, this is now already quite very much part of our healthcare. This is a family with Huntington disease that I first saw in South Africa in the late 70s. In those days, these were families of mixed descent, disenfranchised by the apartheid system which they find themselves in, but also disenfranchised by this terrible, incurable disease. A disease that affects them in midlife, leads inexorably to movement disorders, depression, dementia, and death, usually 18 years from time of onset. And the tragedy of this disease is that for every person affected, each child would have a 50% chance of really suffering from this illness. And for many of these families, they lived at risk right from the beginning, knowing their risk. And for some of these children growing up, being at 50% risk was a tremendous burden. They chose not to get married. They chose not to have children because the uncertainty was too great. And here's just some of the family members of people we looked at. And this is what gorgeous looked like. And so when you met these children early on, in fact, I learned about their humanity and very quickly wanted to do something to have impact on this particular illness. And so people continue to write letters to me about the impact of this illness, the fear, the terrible feeling of losing your mind, dementia, which is such a terrible illness. You know, we have the cost per year in Canada right now is about $15 billion. By the year 2025, it's estimated it will be $150 billion. We're looking at a major increase. Huntington disease represents a tractable form of dementia, which is likely to lead and may lead to approaches to treatment more broadly. Well, the first test, prenatal test for Huntington disease was done here right at this university in 1987, long time ago. And this was done here because, in fact, there were some people that wanted to make sure and know that they were not going to develop and have a child with this particular disorder. At this point, all you could do is you could do prenatal diagnosis, do some genetic tests, and predict with reasonable certainty. By this I mean not 2% increase, 3% increase, but 95, 97, 98, 99% likelihood that this child would develop this terrible disease. Since then, the gene's been cloned. We now can predict with complete certainty whether you will get it or whether you won't. 100% certainty, black and white. And many people are participating in testing, but not everybody. And for them, living with uncertainties is hell. Living with uncertainties is needing to know whether I need to, as I plan for my retirement, hire a motorhome or hire a nurse. I want to know that. And these are people that are choosing to know. And so this particular study started in Canada, and as a Canadian study, very unique. It was unique because it was able to cover the country. And this is a theme that I just want to end off with tonight, which is a Canadian story, really. A Canadian story about working together, sea to sea, coast to coast, people putting all their individual goals in towards a national goal that we need the critical mass to answer some fundamental questions. It's been done many times. But we've done it in Huntington disease and also in adverse events we're going to talk about just in a moment. And when you look at this was people across the country working together to define whether testing could be delivered in a safe and appropriate way, preventing harm, and whether this is something that we should do as part of the healthcare system. And in a sense, this was a public embrace. There was an embrace by the Canadian physicians. The families participated, and Canada became a leader worldwide in developing the guidelines that have now led to predictive testing worldwide. Today, in every developed country of the world, predictive testing is routine, just like you test for your cholesterol. It's part of healthcare, it's paid, and a lot of this emanates from the wonderful collaborative spirit of Canada that led to these guidelines, where we developed the ways and approaches to develop this in a safe and effective manner. And today, of course, also, we spoke about prenatal testing. Well, today you can actually have testing and actually be sure, even before you're pregnant, that you're going to have a child who will be free of this disease. Truly predictive medicine. And this young man, Jeff Carroll, he's been in the news. He's a graduate student in my lab. He was a soldier in Kosovo, American Marine, learned that his mother died of Huntington disease, dying of Huntington disease, came to get tested, found out he carries the mutation, and then decided to do graduate studies to work to prevent the disease that would kill him if there was no cure. At the same time, he wanted children, and he was able to do testing of embryos and then reimplantation of embryos that did not contain the mutation. And so this essentially, and now he has twins, essentially, and he's able to have children that he knows will never develop this disease. And the key thing is to try and prevent ways to do this and prevent this disease for the future. And there's a lot of hope. There are new kinds of approaches that are coming that provide lots of hope for these families. And in fact, in mice, the disease has been prevented. But of course, there's a big bridge, a river to be crossed in going from mice to humans. And the latest medical breakthrough for patients is not there yet, but there is tremendous progress. So Huntington's disease is one example of the personal genome revolution. that has provided tremendous opportunities. It's leading to new approaches to therapy and represents truly a remarkable opportunity for those people who want it. It's been a vanguard for learning about testing, issues of discrimination, stigma, all kinds of issues. But remember, in this disease, when you finish testing, it's a new kind of person in a sense. It's a person who carries the mutation for a disease with 100% certainty that they will get it. At the present time, no cure, so they will get it. So they're not yet ill, but they carry the mutation. They're destined to be ill. They're predestined to be ill in the future. And yet, for many of these people, they're now able to make plans for the future, knowing what that future holds. Now, another area that's a major problem, and a much more common one in Canada and around the world, are adverse drug reactions. And it's always remarkable thinking about it. The fourth leading cause of death in Canada and the United States. 7% of all hospital admissions. And in fact, almost 50% of all health products have really serious adverse drug reactions. Not to say that they're not excellent breakthroughs, but adverse drug reactions are very, very common. And the question is, well, is it likely that genetic variation could in fact play a role in determining the susceptibility to this? And this has been in the news tremendously. If you go to Vancouver General Hospital, 12% of all admissions essentially have adverse drug reactions. That's why they're going there. So the ER visits, very common. And in fact, this goes on all throughout Canada. About 1 in 8, 1 in 9 ER visits are medication related. And the issue that's happened now is that with the success in treating cancer, if you look at cancer, most children are beating cancer. This is a big change, dramatic revolution in cancer therapy. 82% of children are beating cancer. But the real problem today is that these children are surviving cancer, but in fact developing many of the sequelae and side effects of cancer treatment. Today, if you go to Children's Hospital here in Vancouver, 1/4 of all the hospital admissions are not due to the cancer, not due to the complications of the cancer, but due to the complications of the treatment for the cancer. So these drugs that are being used are very serious, and these are children then suffer adverse events much later in life, and this represents a major cause. 75% of these cancer survivors have a serious ADR. So we set up to really, and we recognize that much of this may be genetically determined. Because when you take a drug, how it's metabolized, how it goes across your intestinal mucosa, how it's taken to the liver, how it's broken down, how it's excreted, how it circulates, much of that is determined by genes at critical junctures throughout the body. And variations in those genes influence the activity of them. And could certain variations be associated with some of these side effects? So that was the question we had. And together with Genome BC initially and also CIHR, we were able to start a program that, again, in the same spirit, started here in BC and then spread across the country. We recognize, it's remarkable, when you go buy a pair of shoes, you don't just buy a pair of shoes, and you recognize that we're all different. And yet when we give patients drug, cancer, therapy, whatever it is, therapy, we don't end up looking at the patient to define the dosage and the appropriate way to treat that patient. So we also wanted to focus in the most vulnerable population, and that was children. Another area that's really quite remarkable is close to 75% of all drugs being offered to children have never, ever been tested in children. Children are our ultimate guinea pig when it comes to drug therapy. And in the United States and Canada, we have a massive number of children dying every year due to ADRs. And in the past, we used to think about it, and of course, the number of hospitalizations of children with adverse drug reactions is very high. And the reason we're thinking about children is that most times we think of children, we say, well, children should be treated just like little adults. If I give you in this audience 100 milligrams of a drug, and a child is 1/10 your weight, therefore I should give the child 10 milligrams of the drug. But in fact, it's not quite like that, because children metabolize drugs differently. It's not just about weight, it's how you and those metabolic enzymes that change the distribution of the drugs are very different at different stages. So it's not just about weight, but it's rather about understanding development and understanding metabolism in children. And so we set out to do this initially in children. We worked with a big surveillance network, again, quite remarkable. When I tell my colleagues around the world about this, that 2,300 pediatricians We're following a protocol to determine adverse events in children across this country. This is 95% of all pediatricians in Canada. It's really remarkable, all joined together across this country. And as part of this, we also recognize many of these children are going to hospitals. So we set up a hospital active surveillance network. And you can see it going again, coast to coast across this country. And somebody starting in Vancouver, hired by us across this country in an effort to ascertain these children, to get the appropriate consent. And let me tell you, getting consent across the country for this took a long time, because every hospital had to show how they were more ethical than the one next to them. And whatever Toronto did, everybody wanted to do differently. So we had to change, and this took a long time getting it through, but finally this was approved. And now we also have this incredible network of cancer centers across this country. 17 cancer centers in Canada joined together to address the problem of preventing some of the adverse events for children suffering from cancer. Children disenfranchised by cancer and then having terrible side effects to these drugs. And I just want to, so this is this new pharmacogenomic network of drug safety. It's based in Vancouver, but it crosses the country, and it's been a remarkable and collegial. And let me just tell you, I'm going to give you 2 examples, and then I'm going to finish. So codeine. How many people here have ever taken codeine? Pretty much everybody in this room. How many people have felt drowsy taking codeine? Quite a lot, maybe 30, 40%. Well, that's a serious side effect, that drowsiness. And that drowsiness is often due to genetic variation in you. Some people get drowsy and some people don't. And in fact, till very recently, the American Academy of Pediatrics said that breastfeeding of mothers who take codeine was not a problem. It's no problem. Until this particular case occurred. And this was a young mother, a lawyer in Toronto, who had a baby, not a complicated delivery, but took Tylenol 3 with codeine, essentially, and she became very drowsy. And the codeine was dropped, and her infant also became very drowsy, and by day 13 the child died of respiratory failure. The mother had been very attentive, because as the child had poor feeding, she kept the breast milk. The morphine in the breast milk was dramatically elevated. Furthermore, the child had lethal effects of morphine. The tragic situation is the mother had killed the child through breastfeeding. Essentially, inadvertently, the child had overdosed on codeine. But this was very unusual, what had happened. And so this mother had a particular genetic variation. She had a duplication of a certain gene. She had a doubling of a certain gene, a variant. And the accumulation of this is that she metabolized codeine too fast. And a result of that, the codeine essentially metabolized, crossed the breast milk, and actually went into the blood-brain barrier and created drowsiness in her, and in fact drowsiness in the child, till the child stopped breathing. And the reason the child stopped breathing is children have no defense against opioids, against codeine. Children do not have the enzymes that break it down at all. So it goes straight across the breast milk to the brain, and that's what happened. And then the question became, wow, is this a cause for sudden infant death in children, and is this some of the reasons why children die during breastfeeding? Well, we ended up looking at this, and we realized that, in fact, if you look at codeine, 52% of all mothers in Canada take codeine after pregnancy, after delivery. And furthermore, if you look at those mothers, close to 1.5 to 2% of these mothers have this genotype, have this genetic variation that would predict that their children are seriously at risk. It was this knowledge, just this one case report, and front page Global Mail, and then that led the FDA in the United States to immediately change the label and issue warnings for codeine warning by nursing mothers. The Canadian Public Health and Health Canada also did, and this has now very much changed the way we think about codeine in breast milk, and we now have an ongoing study to define the cost effectiveness of screening mothers during pregnancy for this variation in an effort to define who should and should not get codeine. It's got even worse. This was just last year and published in the New England Journal, where children too, when they're very young, after tonsillectomy, can die during codeine, and it's often been used now because, and this is thought to be due to hemorrhage and other complications, but in fact codeine is the reason it's fatal. Children, again, do not metabolize codeine very well. So this has led to a whole new program for codeine prevention, a way of screening mothers and screening children in particular to know if they're going to have adverse events. If the test is cheap enough, this is something that could be done for a few cents and truly could be personalized matter for people who should not or should take codeine. And I'll give you one other example. This is just published. What's interesting is that 1 million patients a year take this drug for cancer, cisplatin. It's the commonest drug used in children, and it's a very common drug used in adults with cancer. The problem is that in children, about 40% of children go completely deaf on this therapy. And yet, this is, so in adults it's a little higher, it's a little lower, 10 to 38, vary. But at Children's Hospital now, when we went back through those records, close to 40% of children have developed grade 3 or 4 deafness. That means permanently deaf. That means they need either a cochlear implant or they'll need hearing aids for the rest of their lives. This is very, very serious. And furthermore, the younger you are, the more likely you are to have the side effect. Well, there are alternatives to cisplatin. They're more expensive, but they are shown to be efficacious. And so how do we decide who gets cisplatin and who gets the more expensive drug? So we've now been studying this in an effort to answer this. This is what happens to those children. They get severe hearing loss. They require intervention or grade 4, which basically means they're totally deaf. And the cost of this per year for a child is, $40,000, $50,000 a year in terms of extra help beside the psychological burden for life. Well, is there a way to prevent which children will go deaf and which children won't? And I'm pleased to say is now we're able to predict with close to 100% certainty. So this is no longer an increase from 4% to 4.2%. This is an increase from 0 to 98%. We can predict with 98% specificity now which children are likely to go deaf on cisplatin. In other words, if you have this variation, the odds ratio are no longer 1.1 or 1.2. They're in the 40s. And in fact, the specificity of this is very high. You don't pick up all cases of deafness, but it's a very high predictive test. And so this is, again, very exciting in terms of being able to use this and say, well, you can take cisplatin, you won't have the deafness, whereas other children should go into other therapy. BC is taking a leadership role in trying to think about the incorporation of this into therapy. And so they're working with Genome BC and other partners. We're looking at ways to both validate it, think about a deafness prevention program. We also are looking at delivering this test through the test that is now available and delivering this test for people taking cisplatin through the 6 regional cancer centers in BC and looking to determine the outcome associated with this. So this is very exciting. We recognize when we look at all these drugs that no one side does not fit all. Some drugs are effective, some drugs don't work, and we want to try and re-stratify patients, reduce healthcare costs using this, using some screening, and bring genetics and genomics into everyday practice. So will knowledge of genetics revolutionize healthcare? In some areas, it will. In the area of pharmacogenomics, I think we're already seeing some changes. But of course, they're still important. We need to have the same clear guidelines for translating anything into medicine and into healthcare, just like anything else. Just because it's a genetic test doesn't mean it doesn't have to go through proof of cost-benefit, proof of utility, proof that there's an appropriate way to deliver this. And this is what we're doing with cisplatin. This was done with the Huntington test. We need to show clear benefits of clinical utility. We need to show that this is cost-effective, that doing the test truly has a chance to contribute to sustainability of the healthcare system, maybe even reduce healthcare costs. We need to know what the psychosocial impact of the test will be. What's the impact on the family, the community? We need to be able to find ways to translate what we're doing to the general public. The problem is there are very few people. Physicians are not adequately trained in genetics. We need healthcare workers. We need genetic counselors in the community being able to have appropriate translation. And we're going to have to think about novel ways to develop and deliver healthcare, not through physicians. It's going to be through nurses, through various other counselors, and we need to also have a responsibility to make sure that these benefits are not only present for the people of British Columbia and people of Canada, but we also have a responsibility to make sure they are globally relevant for everybody who could benefit from these and do our best to influence that. So I think what I'm saying to you is there's much to be excited about. Personalized medicine has arrived. It's going through the process. It's not quite as fast as we had hoped. It's not quite as dramatic as we had hoped. Genes are not us. Genes aren't us. We are influenced by the environment. But we still are able to bring appropriate approaches to using genetics to influence healthcare with the appropriate rigor and vigor without hyping it, by generating the evidence that's necessary, and then finding ways to work within the system in an effort to deliver this appropriately and healthily and ethically to the population at risk. So I think this has relevance for all of us, and I can't wait to hear what Anita has to pull me apart. Okay, thank you.
Thank you, Dean McCormick. I also want to thank Alan Richardson from the philosophy department at UBC in putting this together, and Chris Stover, that we had the pleasure to meet her today from the Canadian Centre for Ethics in Public Affairs and Genome BC. Really happy to be here, especially given the Halifax connection, although right now I think that probably playing a piano concert for me is actually less nerve-wracking than actually doing this response. What I was thinking about is like, if I were in Professor Hayden's class or have a conversation with him, what would I like him to talk about in pushing us in thinking a little bit further in relation to personalized genomics? So Professor Hayden said towards the very end of his talk, personalized genomics, personalized medicine has now arrived. And so I guess my title here is, so what are we waiting for? So if personalized medicine has arrived, why is it that some of us may still be a little bit concerned about the direction that we're going? Part of my job as a bioethicist is to work at some hospitals in town, particularly in relation to thinking about provider-patient relationship. And so some of the things that I want to think a little bit about today is whether the hope or the promise of improvement in how healthcare is being delivered would actually be the case. Or if we have this kind of promise, how do we actually really make sure that this actually can be delivered? And so my question is, this is all that great, so why are we not all rushing in thinking about getting everybody's genome, entire genome sequenced? And so what may be some of the concerns that we have as we move forward in thinking more about personalized medicine, and how personalized is it really going to be? So there are 4 areas that I'm thinking about today in thinking about whether there are some issues that we may still need to be considering as we move forward in Canada or around the world, in terms of thinking about how may personalized medicine actually, as Professor Hayden said, revolutionize healthcare. So maybe cancer is not at this point a thing in the past, but can at least the use of genomics help us to actually think more about how we may be able to prevent cancer and how we may be able to actually deliver better medicine to patients. So the 4 issues that I'm going to say a little bit about are the epistemological issues, economic issues, ethical and social issues, and the ontological issues. So in thinking about the epistemological issues, Professor Hayden already talked a little bit about how there may still be a lot that we don't know at this point. Francis Collins' prediction didn't come true exactly at this point. We may still want to say, well, from prediction to prevention, it may still lead us to better medicine in general. But as the example that Professor Hayden just used in diabetics, the information that we have is not going to be leading us to certainty. So how do we actually make the best out of it? But when we're still in situations where there's still a lot that we don't know at this point, or even with things that we do know about, we don't really know what to do actually with it. So where do we go from there, and how do we use some of the unknowns to help us actually in promoting better healthcare? So there's a lot that we don't know. The genes about variation in height, 12 genes for 2% of the variation in height. So there's still a lot that we are not sure about, because the effects of any single genes on common diseases at this point are still generally small. We're not exactly sure about the interaction of different genes with environmental factors. The BRCA1 and 2, there is a link between those genes and breast cancer and ovarian cancer, but the genes are not the only thing. Many people who are diagnosed each year with breast cancer do not have these genes, whereas there are people who have these genes may not actually develop the cancer. So we may know that they have the genes, but we don't know for sure whether they will develop the disease, and we don't know which particular person will actually deliver that. So there may be correlation, there may be risk numbers that we actually can tell. But unlike Huntington's, will I get it? When would I get it? In many other conditions, it's really a prediction. We don't know for sure. But what I want to think a little bit about, and maybe we can all think a little bit about, is how much power genomic medicine still have. Even with that much uncertainty at this point, it's big business. There's a lot of money involved, whether in research funding, government funding, or commercial industries. So what exactly might that message be? Or what exactly does that tell us about the way that perhaps even science is being actually portrayed in the media and so on? So even when we know, even when we have the knowledge of genetic risks that people may have, and that there may be some information about disease predispositions, the question is still whether that will actually lead to better health care. It has been known for a long time, even without genetic medicine, that various kinds of lifestyle choices may lead to various kinds of conditions. But having that risk information has not necessarily, in many cases, changed people's behavior. So now we know that smoking in general is hazardous to our health, but we probably don't really need a genetic test to tell us whether we are susceptible to lung disease or not. And very often, in many cases, whether you have a particular gene or not, you may still benefit from various kinds of screening, from various kinds of healthcare behaviors. But whether it is genetic or not, risk factors or risk information has not been so well in motivating people's behavior. So the question may be actually, do we think that genetic information is actually going to change that? And one of the reasons why I'm asking this question, or I'm curious about that issue, is that maybe we have to think a little bit about how deterministic or how certain do we need this information to be for us to say that, for example, in Canada, when we do fund things that we consider to be, or that are considered to be medically necessary, do we want to say that certain kinds of technologies have to be certain of a particular percentage before we would actually say that we now as a country should put that in terms of our funding priorities, research priorities, to say that, well, perhaps these type of predictive testing should be funded and other types should not be funded? So that leads to the question about economic considerations. There have now been some concerns, especially with direct-to-consumer testing in various places, where customers, the general public, can go to different companies and buy these different kits, the spitting parties, I like that, so that they can find out about the genomes, they can find out various kinds of risks, supposedly. But now that we may have a group of these so-called worried well people, they are not symptomatic at this point, but they may find out that they have certain kinds of predisposition to having various kinds of conditions. There may be questions about whether having that information in some cases are actually going to pose even more of a so-called burden on the system, because maybe many of these people would actually want to have more follow-up screening, testing, and so on. So if it is the case that, for example, in Canada, that we decide that, well, maybe at this point, the technology and the genetic information or the genetic knowledge is not such that we really have very much promise yet for various kinds of conditions, then the question would become, should we allow people to buy some of these technologies, to pay for them? So then again, it may get to some of the issues about access, issues about fairness, and so on. So this leads to some of the ethical considerations here. So in terms of insurance coverage, cisplatin was being used as an example earlier. So we may want to ask questions about, is it legitimate, for example, for insurance companies to say that, well, before we actually decide whether this particular patient, her or his medication should be funded or covered by the insurance plan, that we would want to require that this patient's genome is being sequenced or various kinds of genetic tests are being done in order for us to say whether we believe that this particular medication is actually a legitimate one for this particular patient. Now, in cases that we may have almost 100% certainty, that may make a lot of sense. But you may ask questions about other kinds of conditions. HER2 was being mentioned earlier about Herceptin for women with breast cancer. Should we say that, well, actually, we won't fund Herceptin for women who have not got the test done, and only those who have got the test done and show positive in certain areas, that their medication would be funded? So in some ways, in some cases, it may make scientific sense. But in others, if we don't really have very high certainty, then we may have to ask some of these questions. Now, of course, once the genetic information is being gathered, then we also have to ask questions about what do we do with actually that information. If insurance companies can get their hands on that to make decisions about one kind of medication, should they be allowed to also use that for other kinds of medication or for maybe life insurance purposes? This has, of course, raised a lot of questions in people's minds about whether some insurance companies may be able to so-called discriminate certain patients based on their genetic profile. Stratification was being mentioned in relation to disease, but we may also ask a question about whether there could be also social stratification based on a person's genetic profile. And so in some cases, then the type of discrimination that people may at least perceive to be happening can lead to perhaps in some ways even more distrust in some of the way that we think about the genetic information. People may say, well, actually, it's not so much that I don't trust the science, but when we don't have all the information, when the knowledge at this point is still limited, but as being used for various kinds of policies, for industries, for companies, or perhaps even for the government, then there may be more distrust. And this, I think, is also linked to the ontological question in terms of who we think we are. The example that I was talking about in relation to a person who may be adopted and believing that actually going to the direct-to-consumer company, the 23andMe or others, to find out more about the genetic information, if they think that this is actually going to tell them who they are, then we may have the question about whether we're assuming that who a person is really completely just depending on the person's genome. Professor Hayden mentioned that, now many people would say, well, everything is genetics, right? So even though, was it the Time magazine that you said, why your DNA isn't your destiny, right? So it maybe some magazine covers now say that it's not really the only thing. That's not your complete makeup in terms of who you are, in terms of your personal identity. But it does seem like we're getting very mixed messages. On the one hand, we're pushing a lot in terms of genetics. We're thinking that personalized medicine is very much based on one's genome. But at the same time, we also are trying to say that, well, but wait a minute, that really isn't the only thing. And so some people may be actually really getting confused about what kind of message should we take from here. There are more and more companies actually developing these tests. The cost is actually getting lower and lower. It almost seems like it would be silly for us if we have access to some of these technologies to not actually utilize that. But at the same time, we are also trying to tell people that, well, even though genetics is everything, but at the same time, it really isn't. So, but again, if it really isn't everything, then it comes back to an earlier question that I asked too. Why is it the case that there is so much power, or that many of these industries or companies have that much power in actually changing the way that we actually think about healthcare? In various ways, pregnancy being mentioned earlier about prenatal testing, some people may also ask the question about whether some of these genetic tests now are also changing the way that people think about pregnancy, thinking about childbearing, child rearing, and so on, or what it really means to become a parent, what it really means to actually have a child. It may also lead to various kinds of questions about who actually gets to define what the particular information actually mean, in terms of also, for example, defining who we are, and so on and so forth. The who we are, I think is an interesting question, especially when we look at a particular drug that came out in the mid-2000s, the Bidel. This was developed in Minnesota as a drug that was actually for the African Americans, for the heart disease. The Wall Street Journal, the headline there says the FDA panel backs, I can't actually even see what it said, use of heart drug in black patients. And then USA Today had a very similar article about that. And since that time, the idea is, well, actually, the company in the end got the patent to market this drug purely to African American patients. even though the scientists behind this actually also thought that, well, this would very likely work for patients from other races, right? And so there have been questions about how do we think about racialized medicine. One camp of people thought that actually this kind of racialized medicine was really helpful. One, not only are we now paying more attention to certain populations that have been actually left out and marginalized in medical research, genetic research, but now, especially when we are targeting more certain populations, maybe they're actually more prone to talking to the physicians about their health care. But some of the concerns that came up from that too is also how do we think about the so-called racial identity? And so what does that really mean to say that this particular drug is actually good only for black people versus other people? The trial was done on African American people, even though the same drug was actually, this particular drug was really, 2 drugs that have already been used have been on the market. But by marketing this to African Americans, the company was able to extend the patent for much longer. So there were questions about, at least, whether science and I guess business initiatives were mixed together, and that at least makes some people very skeptical about some of the claims here. And of course, it also makes some people very concerned about whether we are once again talking about race as a so-called biological concept rather than the social construct. And I know that we don't have time to actually get into that kind of debate, but it does actually raise various kinds of questions in terms of how one gets to actually define herself or himself. I mentioned in an earlier slide about how would that information be actually interpreted, who would actually come up with the meaning about this particular kind of interpretation. And so in various ways, and we have to ask questions about whether sometimes the so-called genetic differences, especially on so-called racial terms, may also be used to explain or perhaps even justify various kinds of health disparity. So if it is determined that this particular person is of a particular racial heritage, there are tests for that too, like genealogy and whatnot. And so how does that balance from the way that people may self-identify with their own identity? Genetic tests have also been used, or at least have been, I don't know if accused is the right word, but concerned in terms of how it actually changes or perhaps even give meaning or determine meanings of disability for patients or the word well that I mentioned earlier, or for other patients who are asymptomatic at this point, maybe asymptomatic for the rest of their lives because of various kinds of genetic susceptibility. Again, there may be questions about how are we actually going to define these people. Can employers, for example, give genetic tests to determine the risk that various people may have for, let's say, carpal tunnel syndrome, which have been used in a particular real-world case in the U.S., or other kinds of industry to say that we want to try and test whether this particular person may be, let's say, susceptible to depression and so on. And even if the person does not actually ever exhibit any of these symptoms, if the tests are shown to say that this person has the susceptibility or predisposition to some of these conditions, is it going to define the person? Is it going to define the person, for example, as being disabled, even when the person may actually be asymptomatic? So even though we may say that, well, with the DNA or genome sequencing, we can say that, well, there's never been anybody like you, or there's nobody that's going to be ever like you. But if that's the case, are we, again, just talking about it in relation to just a genetic identity? In many ways, we may say even identical twins was being brought up. In many ways, people don't think about them as, the same person, right? But somehow, are we just talking about it only in terms of the genetic information here? So to wrap up, do we think that actually all the genomic information or genetic information that we're thinking about here can really help us to make medicine actually more personalized? As I mentioned earlier, part of my work is in hospital and working actually at the bedside with patients and providers. And very often the way that when I think about whether some particular treatment or particular encounter is actually a personal one or not, it's not just about the drug information. It's not even just about the treatment decisions. Very often I would at least have to assume that all the information, all the treatments that are being given are actually uniquely available or uniquely suitable for this particular patient. But very often, what patients really care the most about is the interaction that they have with the providers they have. Whether the provider actually care about the concerns, the needs, and other contextual factors that they are facing. Of course, again, not to say that the treatment plans are not important, but that is only one part of the experience that they're going through. So even though I don't want to say that genomics are not important in thinking about the future of healthcare, but we may have to sometimes take a step back and to ask ourselves whether sometimes an overemphasis on genomics or genetic technologies may take away some of the more personal aspect of medicine. And so in some ways, that maybe it actually takes away the way that we think about how healthcare is really more supposed to be holistic, caring for the whole person, not just about the fragmented and maybe even the invisible part about genetics of the person. So that we may have to actually, especially as we get more and more into the age of genomic medicine, that we have to think a little bit more about what exactly personal healthcare is actually for each particular person. If genomic medicine can actually help physicians who are actually at this point not very knowledgeable in genetics to actually give better information, to actually direct the patients in better ways, to have better communication about the medication, behaviors and lifestyle choices and so on, I think that probably we will all be more for it. But if genomic medicine somehow may actually divert attention away from that kind of personal attention, then we may have to worry about whether actually the so-called personalized medicine is actually the most impersonal. Thank you.
I don't think there has to necessarily be a dichotomy between genomic medicine and taking care of the whole patient. It's just one factor that influences. So it's not as if you're getting some DNA, it's no different to any other result. It's a result that influences healthcare. It predicts something. But you always, as a physician, have to care in the whole patient. And there's it's not just that this suddenly is going to divert this physician away from taking care of the whole patient. So I think that's sort of just not necessary. It's not a necessary outcome. On the commercial side, of course, it is interesting, and I don't think this is a free country. Many people choose to have healthcare in many different places. Those who are wealthy go down to the United States and other places. Whilst there may be good and bad associated with that, I don't think we as a society, the social cost of legislating that people can or cannot pay for healthcare as they choose would be profound, the social cost. And so I don't think we're going to be able to say to people, it's not going to be allowed for you to go and get some tests from 23andMe. What we can say as physicians and as scientists is, what you're going to learn there is not going to be all that certain, not going to be all that helpful. And if you want to spend $400, by the way, this week there's a special on with 23andMe, $199 if you do it by Friday, to get a certain number of SNPs looked at. And if you do that, but we might want to say that $199 would be better spent joining a gym. There's also a special at Gold's right here, so you could join and get a special rate, and that might be better for your health as well. And it's interesting, there was a little study done by Jim Evans from North Carolina, and he took the same blood sample and sent it to 4 companies offering the same test, more or less the same test. And out of the tests given, about 40% of them were completely divergent. In other words, one told him he had an increased risk for diabetes, the other one a decreased. This was a guy with a lot of hair. They told him he had a high likelihood of going bald, the whole thing. So the real problem isn't so much the technology itself, but it's the certainty of the information. It's still very early. And I think what we can do as physicians is direct people to the uncertainty around this. And then in terms of incorporation to healthcare, I think this has to go through the appropriate rigor. You have to show that there's truly scientific evidence. We have to be based in the evidence. And then from that, you have to show that there's cost benefit. And then you have to look at this as the prioritization in terms of all the other healthcare needs. And you have to argue why and provide the benefits as to why this is cost effective, taking the sustainability of the whole healthcare system into account. So this has to be put into its context. There are some family physicians who don't like the word personalized medicine. I understand, because they say that's what we do every day. We provide, hopefully, personalized medicine. But I think this is referring just to the technology, and so the terminology may be a little bit over and may, in a sense, suggest that what everybody else is doing is impersonalized medicine. But I don't think that's the spirit in which that name was coined, and I had nothing to do with it. Okay.
So, can I open the floor? Is there anyone who would like to make a comment, a question, a harsh retort?
Foucault's sort of great lesson from the birth to the clinic, which was that changes in practices, pedagogy, even things like record keeping within medical institutions change the way in which we think about things like human nature. And this kind of came out from Anita's discussion of ontology. So now we have sort of technomedicine, we have evidence-based medicine, there's capitalization in medicine, and genetic medicine. And so I was also sort of thinking about how Nick Rose kind of talks about genetic medicine as being part of the molecular gaze, and what that molecular gaze sort of does in terms of establishing certain kinds of things as the appropriate objects of exploration. And again, Anita kind of touched on this, but one of the things I wanted both speakers to comment on was how the sort of molecular gaze shifts patterns of thought, or in some cases it doesn't shift patterns of thought. So what we have potentially is a carrying on and even furthering of Enlightenment narratives of positivism, individualism, and rational mastery, and what that sort of has to do with respect to situating kind of science in as part of nature versus culture.
I guess maybe I'll just make a brief comment about the shifting the pattern of thought. I guess that's part of my question, right? And I'm not sure whether it's part of my concern yet. I want to say it is, but I know that I'm probably also in a room full of scientists, so I don't want to say that for sure, because I think that the shifting the pattern of thought could be a good thing. But I guess I'm just a little bit more cautious in terms of who gets to define the direction of thought that we should be taking, right? And so, I use Baidu as an example there, but especially given the way that the funding is actually going, and I guess I'm a little bit concerned about that paradigm. I guess I'm a little bit concerned about the shift in terms of thinking about what actually healthcare means. I do agree with Professor Hayden that I don't think that, for example, that genomic medicine necessarily have to mean impersonal medicine. But I guess when we put so much focus, when we think about funding, and I was just looking at CIHR funding the other day, when we think about how research is being funded these days, it's not in clinical medicine in terms of how medicine is being delivered. As much as CIHR wants to say, for example, that we want to focus more on I can't remember, like the areas in 3 and 4, more about the system of delivery. At the end of the day, it's still the hype. What we see on television or in the newspaper is really more about genetic medicine. So I think that actually does, in some way, shape the way that we think about where healthcare is going.
I again like to say that, firstly, I think as geneticists, often by going deep into those genes, you also understand how it doesn't capture everything. So I think the more we study, the more. I'm not a genetic fundamentalist. We recognize that genes aren't us, but to make the advances that we need to make, it's not to deny anything else, but rather to focus and make those rigorous, serious discoveries that impact on the delivery and development of new approaches to therapy. Yes, you've got to focus down and down and down in an effort to do that. But let me just say also, when we think about all the genome projects across the country, and this started with Jim Watson right in the beginning, there is a defined amount, and every one of these projects has an ethical, a legal, a social component to them. In terms of these, and that's very integral. We're looking at the view of adverse events in families, and we're working with people in Montreal and social effects within families, and how is the concept of the family altered by this. And I think that one's able to, and certainly when we look at the large genome projects, other dimensions besides the fundamental genetics are very much incorporated as part of this. And this is very much a multidisciplinary team working together, I'd say, in remarkable ways. And we're learning from each other and finding colleagues and in ways and informing the way we think and the way we talk in a profound way. I, for example, run my lab. I see in the audience I have a grad student here from interdisciplinary studies. But we have detailed fundamental scientific meetings to which the people doing social science research are present. Then we have these individuals, and there's so much that we learn by bringing this all together in one place. And I think, so our gaze is deeply enriched by exposure to each other's gaze, and by sharing of that in an atmosphere of collegiality and cooperation and rigor. The other thing I would say is that just because it's not molecular research doesn't mean you can't bring the same rigor to every other form of research. And I think that's part of the challenge too, to bring the appropriate rigor that says, is this real or not, and is this, and how do you bring that rigor to other forms of research that demand it? Because we need to know the impact and the way it's affecting our image of who we are. But we need to bring appropriate rigor and attention. And care in terms of those interpretations. And this is improving all the time. So thank you for that question.
I'm going to ask you a question before the next one is on. When you were talking about people who choose to live ignorance, even when they can be informed, whether they're a very person. I often think of that as a sort of release moment. That is it really always better to know?
Well, in fact, the vast majority of people would say no. Yeah. Most people choose not to participate in testing. You know, before the tests were available, we did all these surveys, and many people around did these surveys. About 80% of people in the world, all over Europe and the United States and actually even parts of Asia, said they would want to participate in testing. The research that was undertaken, they didn't ask when they'd want it. So then the test became available, and today what's interesting is British Columbia in particular, but Canada in general, has the highest number of people in the world that is participating in predictive testing. So, but still